Okay, we're going to talk about alcohol and how it relates to dementia and to cooking oils. These things are all connected. So we'll have to start out with alcohol metabolism, and then we'll show how that relates to lipid peroxidation, brain damage. And the purpose of this talk is just to make it clear that you could very easily reduce your risk of dementia by lowering your intake, and I recommend avoiding altogether these cooking oils and alcohol. And one of the interesting things that will come out of this talk is how common and powerful alcohol and cooking oils are to cause brain damage. Okay, so starting out with the metabolism of alcohol. So remember, for ethane is a 2-carbon alkane chemical, and the key point is ethane is the root of the word, and ol means alcohol. So ethanol is ethyl alcohol, and that's what everyone means when they say alcohol. So 2 carbons and then a hydroxyl group, and hydroxyl group is also an alcohol group. It's metabolized in the liver by ADH. That stands for alcohol dehydrogenase. And during that reaction, NADH is produced. So the hydrogen is taken from here and put over to here. Then an aldehyde is made, and that is called acetaldehyde. Ethanal, the Al indicates an aldehyde. So that just means the carbon has a carbonyl group on it, a double bond to oxygen, and we now, again have a hydrogen over here. This is toxic. This acetaldehyde or ethanal, it's the same thing. That's what causes the symptoms of a hangover. That's what causes the symptoms of Asian flushing syndrome, when they turn red and they're tachycardic and nauseous and can vomit. And the persons who get that are persons who lack this enzyme, acetaldehyde dehydrogenase, ALDH. This reaction step is irreversible. It also produces NADH, and that's an important theme, that a lot of NADH gets produced by this alcohol metabolism. This acetic acid or ethanoic acid is not toxic. It's a carboxylic acid because the carbon has a double bond to oxygen and then has also an attachment to a hydroxyl group, an alcohol group. Okay, we're now going to go into a little more detail here. So again, the alcohol enters the liver cell. In the cytoplasm, the reaction we just looked at, alcohol dehydrogenase converting it to acetaldehyde, same thing as ethanal. That acetaldehyde will then go into the mitochondrial matrix inside the mitochondria, the center of it, and ALDH is located there, and it will convert it to ethanoic acid. So this is going to produce NADH in the mitochondria. So you're going to have accumulating NADH in both the mitochondria and in the cytoplasm. And what that means is there's reactions dependent on that. They're going to start to, their direction is going to run backwards because of this accumulation of NADH. We'll get to that in just a moment. When a person drinks a lot of alcohol all at once, the liver cytoplasm ADH system cannot handle it. And the extra alcohol is then passed into the endoplasmic reticulum, also called a microsome. And that has a special enzyme called the cytochrome P450 CYP2E1. So don't worry about the name. You don't need to know that. What you just need to know is that overflow alcohol is metabolized on a separate pathway. And this one generates NAPD, but the real key thing about it is it generates reactive oxygen species because the cofactor includes oxygen. Some of those reactive oxygen species will damage the cell membranes of the liver cell. It's called lipid peroxidation, and that will produce toxic aldehydes. So MDA is from malone D-aldehyde, 4-HNE is 4-hydroxynonenal. That's really that's the one you want to know, 4-hydroxynonenal. And it's typically abbreviated 4-HNE. And that will bind to proteins and decrease their function. It'll also bind to DNA and damage DNA. So that's a key concept. It's going to keep coming up, toxic aldehydes. And so the point of this slide was the more a person drinks, the worse it gets. A tiny amount, the body can handle it reasonably well. I still think alcohol is toxic. It's like throwing a monkey wrench into your metabolism. Wait till you see how much worse it gets. But still, a small amount, the body's got a lot more ability to handle that. Large amounts, it just gets worse and worse, the side effects. Okay, now here's showing a couple chemical reactions in the body that are very important and end up running backwards when a person drinks alcohol. The first one is glycolysis. So we're in the cytoplasm of the liver cell and other cells, and this is an important glycolysis intermediate. The substrate here is 3-phosphoglyceraldehyde. And normally it will run in a forward direction to 1,3-bisphosphoglycerate. Okay, and the point is that this is right in the center of glycolysis. one of the most important metabolic pathways in the human body to extract energy, most commonly from glucose. And because there's so much NADH accumulated, and the NADH is a cofactor for the reaction, this reaction will run backwards, and that's bad. You can't get energy from glucose if glycolysis is running backwards. So drinking alcohol can cause a pretty rapid drop in somebody's performance, let's say for any type of athletic thing or cognitive thing. So that's the first reaction that's damaged by drinking alcohol. The second one is the conversion of lactate to pyruvate. So when you want to be able to convert lactate to pyruvate, let's say after exercising, it's a way you can generate more glucose and get some energy back. But you can't do it here because the NADH accumulation that happened from the initial steps in metabolizing alcohol are going to favor running this reaction backward and producing lactate from a beginning substrate of pyruvate. So that's bad. So that's going to make it so you can't run gluconeogenesis. And now a person can get fasting hypoglycemia, so low blood sugar levels, because they can't get enough energy from the glycolysis running the normal way. They can't make glucose through gluconeogenesis effectively because of this reaction running backwards. A lot of alcoholics are malnourished to begin with. Sometimes you'll have a diabetic who gave themselves a little bit too much insulin and drank alcohol simultaneously, and they can become quite hypoglycemic. In addition, metabolism is damaged in Krebs cycle. Krebs cycle reaction, normally malate proceeds to become oxaloacetate, but that reaction also runs backwards. So you can see what a metabolic disaster alcohol is. You're running glycolysis backwards, you have a block in your gluconeogenesis to generate glucose in the liver, and you have a block in your Krebs cycle. I mean, that's a disaster. We talked about how diabetes creates a disaster in the skeletal muscle and subsequently in other locations running electron transport backwards. I don't think I've talked yet about cancer metabolism. Cancer will run Krebs cycle backwards. So those are three disastrous things. So alcohol, diabetes, and cancer, they make metabolism go backwards. Trust me, that's a disaster. You don't ever want that. Okay, just a couple thoughts on high NADH relative to NAD plus ratio. So we talked about that. The NAD plus is a cofactor. Pretty much all your dehydrogenase enzymes, the ones that are relevant to us, they're all going to have NAD plus as a cofactor, leading to NADH in the normal direction that we talk about for these reaction pathways. So like I said, alcohol is like throwing a monkey wrench into metabolism. We talked about the cytoplasm enzymes. Oh, the other point is that because you're going from pyruvate to lactate in large amounts, lactate is going to accumulate, and that leads to a lactic acidosis. That's not good. You know, remember we talked about cancer preferentially grows with an acidic milieu, surrounding environment. So when you're drinking alcohol, you're producing more lactic acid. And so you want to optimize your terrain, if you will, your tumor environment, make it more alkaline, not acidotic. So alcohol is a major carcinogenic substance. And I can tell you, I've seen tons of patients with alcohol-related cancer. Don't get me wrong. They're much worse off if they're eating animal protein and other things and smoking cigarettes too. But alcohol, major cause of disease. It's routine. I'll see a patient with a disaster of a disease and it's from alcohol or alcohol contributing to it. Alcohol is a major thing that causes premature death in this country. So what else happens? Well, you accumulate fat in the liver because you can't run the energetic pathway so well and you've got so much of this stuff coming in all of a sudden. So alcohol strongly predisposes to fatty liver. I see tons of fatty liver every day. It tends to be worse in the alcoholics than the person who's just eaten processed food and junk food. I mean, don't get me wrong, they can get a pretty severe fatty liver as well, but in alcoholics, they often get a severe fatty liver. And that fatty liver over time is associated, like we just spoke about, with the endoplasmic reticulum. Sometimes it's called the microsomal ethanol oxidizing system, producing the reactive oxygen leading to scarring and tissue damage in the liver. So over time, fatty liver can progress to extensive scarring in the liver, fibrosis, and that leads to cirrhosis, end-stage liver failure. Also, because you're not effectively running Krebs cycle to burn fat for energy, you'll get some of that extra acetyl-CoA made into ketone bodies. So you can get a ketosis, and that can lead to a worsening acidosis, all of which, again, is creating a more acidotic environment, which is something we don't want to do in our blood. And if you really drink a lot of alcohol all in one sitting, like binge drinking, again, you can overwhelm even the endoplasmic reticulum enzyme. And there's another enzyme, catalase, that comes into a minor amount. But the bottom line is you can overwhelm the liver's ability to detox. And even a person with perfect enzyme systems can still start accumulating these toxic aldehydes. So drinking alcohol is a really bad idea, especially in large amounts. Okay, other problems for alcohol. The so-called benefit of 1 or 2 alcoholic drinks per day has now been contradicted. The data has been reevaluated, and it turns out that they were including persons who quit drinking alcohol because they were sick in the category of non-drinkers. So these people, for whatever cause their alcohol did to make them sick, that could contribute to why they died early. So the point I'm saying is it appears that the previous idea of 1 or 2 alcohol drinks per day is not really that true. I always thought it was bogus. I mean, like, how could alcohol be good for you in any way whatsoever? I never believed it from the start. And you have to be that way. A lot of people say, well, I want to go by the data. Well, you know what? A lot of times the data is bogus, and you have to use your brain to think through it. Does this make sense? Is there a plausible mechanism? Is there a big epidemiological study that helps confirm it? So those are good questions to ask yourself when you've got a topic that's a little bit on the debatable side. All right, ethanol is about 7 calories per gram. Just empty calories, major increase in cancers. And a lot of these alcohol cancers are bad. You're pouring this, tumor-promoting substance. It's a metabolic poison, really. You're pouring it over your oral pharynx, your mouth, your throat, your esophagus, gastric, all these ENT cancers. You don't want them. Big surgeries to use to treat them. You don't want that. Esophagus, colon, breast cancer. Okay, so persons who are heterozygous. So this is aldehyde dehydrogenase enzyme. Typically there's an isoform, meaning different subtypes of it. We don't really need to know that, but just be aware of it so when you hear it, you're not surprised by it. And then the asterisk is called star, so star 2. So persons who have that, that's the decreased ability to metabolize alcohol. They also have increased esophageal cancer if they drink alcohol, and let's say they're heterozygote, so they're able to drink a little bit of alcohol. Alcohol also causes leaky gut. So that enables gram-negative bacteria, lipopolysaccharide, to cross the intestinal lining, cause inflammation. That's another topic, but just be aware it causes leaky gut. So if you got an autoimmune disease, you want to avoid alcohol. It can cause pancreatitis, post-pancreatic diabetes, contributes to hypertension, stiffens RBCs, and other problems. Alcohol causes tons of different problems. Okay, just wanted to show a little bit about what lipid peroxidation looks like. So here we've got let's say a cooking oil, omega-6. So you know it's an omega-6 because we're counting from the methyl end. This is the carboxyl end. This is the methyl end. The methyl end is also called the omega end. It's typically marked by a W, which means omega, but it could also be an N for omega. So we go 1, 2, 3, 4, 5, 6. So at carbon number 6, there's a double bond. And then there's always a carbon in between that doesn't have a double bond on it. That's called the methylene bridge. And then we'll come to the next carbon 9. Typically they're spaced about this much apart. So the double bond locations will be 3 apart from 6 to 9. Okay, so this methylene bridge carbon has a loose hold on its hydrogen because the double bonds are pulling on those electrons, and this hydrogen can get plucked off, and when it gets plucked off, oxygen can react with it and come onto the fatty acid and produce a peroxyl, meaning 2 oxygens next to each other. So 2 oxygens next to each other is a peroxyl, and that can initiate a chain reaction that just destroys plasma membranes. Okay, there's some antioxidants. Antioxidants are chemicals that are able to donate an electron and maintain stability themselves, not keep on reacting. And that's usually because they got a whole bunch of double bonds that are, in a sense, like resonating and conjugated, so they can delocalize that electron charge, and that's why they're able to remain stable. So these lipid peroxidation reactions are much, much more common in a polyunsaturated fatty acid. So something like linoleic acid with 2 double bonds. They still happen in MUFAs like oleate, like with olive oil, and they are very, very rare in a saturated fat. So this is one of the few times where saturated fat comes out looking good. Okay, alcohol and cognitive impairment. We're going to get to the Asian dementia. That's going to be sort of like the highlight of this talk. It's coming in just a moment, just so you know. But just briefly, alcohol and cognitive impairment. I look at tons of brains, and I can tell you, alcoholic brains look terrible. They look 30 years older than they are. They're a disaster. Their brain's all shrunken and atrophied, like a shrunken brain. It looks pickled. Sometimes they have abnormal T1 hyperintense signal in the basal ganglia. Once the liver can't function effectively because of cirrhosis, it normally has to convert ammonia into urea, and when it can't do that, that's neurotoxic, the ammonia, and that can cause confusion, hepatic encephalopathy. Really heavy drinking toxic aldehydes can acutely damage brain cell membranes. Alcoholics over time tend to become low in thiamine, and that's a cofactor for pyruvate dehydrogenase, another important enzyme that connects glycolysis and Krebs cycle, can lead to Wernicke-Korsakoff syndrome. I've seen destroyed corpus callosum from Marchia fava, Bignami disease. So alcohol is a disaster for the brain. So yeah, when somebody says, oh, they want to get drunk, they want to get stoned, I mean, it's just stupid. You want to poison your brain? Why do you think that's a cool thing to do? It's stupid. Okay, so here now talking about the Asian issue with regard to alcohol. So normally acetaldehyde dehydrogenase, it's called ALDH2 star 1. So that's pronounced star. Normally a person has 2 copies of ALDH2 star 1, and they get 1 copy from each parent. Worldwide, like we said, there's about 550 million persons who are deficient in ALDH. The abnormal version of ALDH is called ALDH star 2. And so if somebody's heterozygous for that, meaning that they have 1 normal copy and then 1 abnormal copy of the gene, the abnormal version tends to have partial dominance, so they usually only have about 20% function of ALDH, so they really can't drink much. If they're homozygous, that meaning to have both copies, the same abnormal gene from both parents, to inherit that, they'll have no function of ALDH, and they really can't drink essentially at all. They're very prone and rapidly develop flushing, tachycardia, nausea, vomiting, catecholamine release. The key point, though, is ALDH is used for the metabolism of other toxic aldehydes. And that's really the key point of this talk, is that these persons should not be eating any of these cooking oils, the omega-6 cooking oils. They're really bad. And the guy who figured this out was this Setsumori Yamashima. He's a Japanese neuroscientist, physician, and people were wondering, why are so many people in Japan becoming demented? And he figured out the relationship. We'll go into that in just a moment. So the lipid peroxidation aldehydes destroy brain cell membranes, and a person deficient in this enzyme, because they lack a significant amount of ALDH, they really have very little defense against a progressive worsening destruction of the plasma cell membranes in their brain cells and the lysosomal membranes. We'll talk about that, how that happens here in just a moment. Your brain cells are different than, let's say, the stem cells of your gut. Brain cells don't turn over. They keep memories of your entire life. But what a brain cell has to do is it has to recycle its proteins. The proteins within a cell, they become damaged over time. They get glycated. They can have protein adducts from toxic aldehydes. And so they have to be taken to the lysosome and broken down into their constituent amino acids, and then new proteins made from those. And that's a normal recycling process that our body does all the time. And there's something called heat shock proteins. The heat shock proteins, that's just a name for certain properties it has when heated, but they are what helps the lysosome to run its recycling business, so to speak, for the neuronal proteins. So here's a graph just showing that hydroxynonenal, that's the toxic aldehyde from lipid peroxidation and from cooking oils. It tends to be higher and higher levels accumulating in a person's blood as they get older. So older people tend to have more of this. And in general, it tends to be true that older people have less efficient detoxification systems. They have less nitric oxide from their endothelial cells. They've got less endothelial precursor cells. They've got less elastic fibers in their ascending thoracic aorta. Their immune system's less functional. So what I'm saying is the older you get, the more fragile you get. So you need to be more and more careful about your health habits. And this is just one more dangerous thing that's working towards causing dementia. So the smart move is don't eat any cooking oils, not one drop, and you'll avoid all these problems. The other thing about the brain is the brain doesn't just get a little worse, give you a warning, get a little worse, give you a warning. No, you kind of keep getting worse, 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 and then you get a warning of having symptoms, and it's too late. You're screwed. So you want to prevent stuff. Just like a coronary artery atherosclerosis can present with sudden death from a myocardial infarction, you want to prevent coronary arteries. You want to prevent dementia. Okay, so here's this Tetsumori Yamashima, MD, PhD, Japanese neuroscientist. And he started studying, dementia, Alzheimer's dementia. By the way, I don't even like the word Alzheimer's that much because it implies that dementia is usually Alzheimer's. That, in my personal experience, is not true. From what I've seen, vascular dementia is by far the most common cause. And there's a lot of pathways to vascular dementia. When I look at these brain MRIs of tons of demented patients, I don't tend to see the so-called classic Alzheimer's pattern of, atrophic medial temporal lobe and parietal lobe. No, I just see diffuse cerebral atrophy. And I see quite often a lot of ischemic disease, meaning little strokes or so-called silent strokes, periventricular flare hyperintensities. All right, so what did Dr. Yamashima discover? Well, what he found was that the cooking oils were leading to the production of toxic aldehydes, and the persons in particular who were deficient in ALDH were unable to detoxify them and had much more rapid damage and were more likely to get demented at a younger and younger age. And so these toxic aldehydes are made by heating a cooking oil to over 200 degrees. And the bad thing about it is just the processing of making cooking oils, they're often heated over 200 degrees in their processing. So it's routine that they contain, even if you don't heat them up, just from their processing to make them, they already contain H&E, hydroxynonenal. So they're always bad. Okay, and even if it wasn't for that, they'd still be bad if it wasn't for all the stuff we're talking about. They're an unnatural food, like we spoke about that before. Adam and Eve could not eat them. Okay, they're not natural. They don't occur in the wild. We're not made for them. 4-H&E is highly diffusible, can travel all over the body, get in all kinds of location and damaged tissues. In the brains of patients with dementia, so-called Alzheimer's dementia, they find a lot of H&E in beta amyloid plaques, in the neurofibrillary tau-type tangles, and in the cerebral spinal fluid. In Parkinson's disease, they find high levels of H&E in the mitochondria and the Lewy bodies. And basically what Yamashima says is that up to about age 30, people are reasonably good at detoxifying these. If they don't eat excessive amounts, they can have some damage before them. But after 30, they progressively are less able to process these toxic aldehydes, and they're more likely to be damaging brain cells, increasing their risk of early onset dementia. So to go into a little more detail about what Yamashima figured out was that I have a picture of it. His theory is called the calpain-cathepsin hypothesis. So calpain is a protease that's going to cleave the heat shock protein. I'll show you a picture of it in just a moment. It'll make more sense. Cathepsin is a protease within the lysosome that breaks out of the lysosome once the heat shock protein has been cleaved, and then it just destroys everything in the cell and the person dies. So if you want to look at the research paper, here's a reference to it. Can calpain-cathepsin explain Alzheimer's neuronal death by Yamashima, 2016. All right, and Yamashima's got other papers available on there. Let's see. In the brain, it's a heat shock protein, HSP number 70, that chaperones proteins to the lysosome for degradation. So chaperoning just means it binds to the proteins, it delivers them to the lysosome. The lysosome is a recycling factory equivalent of a cell. The lysosome breaks the protein down into its individual amino acids, and those are then sent back out so they can be used to make proteins. Autophagy, auto means by itself, phagy is to eat, so it's sort of eating itself in a sense that recycling. That's the scientific medical word for it. HSP also stabilizes lysosomal membranes because it increases production of ceramide. And it's actually the ceramides, a type of fat, which helps maintain those lysosomal membranes strong so they don't break apart. Very important, lysosomal membranes don't break apart because they're full of very concentrated digestive substances, if you will. The metabolite HNE, the toxic aldehyde, when it's present, hydroxynonenal, it will bind to the heat shock protein, HSP, and that's a really big problem. damages the function of HSP, and in particular, it makes HSP more attractive to calpain. Calpain is an aggressive protease enzyme in the cell cytoplasm. So when HSP is bound to the toxic aldehyde, HNE, now calpain becomes attracted to it, and it will cleave HSP, cut it in half, okay? And now HSP can't do its job of chaperoning proteins, and it can't do its job of helping ceramide stabilize the lysosomal membranes, so the lysosome will break apart, and it will release its inner contents out into the cell, and they will auto-digest the cell, cause the cell to die. So basically, if a person with ALDH deficiency eats these cooking oils, they are very high risk for extensive neuronal brain cell destruction. Okay, and even in a regular person who's got perfectly good ALDH, they get this happening, but it's much worse in a person who's ALDH deficient. And so this was the subset of patients in particular that were at increased risk for dementia, and Yamashima figured that out. Things that increase cytoplasm calcium will also cause increased activation of calpain. And that's a great name for it, calpain. It's calcium activated, cal. Pain, as it causes pain, does damage, big time damage to your brain cells. Okay, and so what I'm saying is other things increase cytoplasm calcium in a brain neuron. And we talked about these in other lectures. You know, there's the potential excitotoxins, MSG, MFG manufactured glutamate, excitatory amino acids, aspartame. All these things are thought to function as excitotoxins in the hippocampus memory center of the brain. A diet low in sodium, you know, basically a processed food diet. High sodium, I meant high sodium, lack of potassium, magnesium. All of those things are going to lead to increased cytoplasmic calcium concentrations. Also SERCA inhibitors. I'm kind of going off on a little tangent here just so you're aware. This stuff all connects. And basically, when a lot of people become demented, they're not demented because of one simple reason. That's why there's no pill that can change this, because there's a whole bunch of little things, and you have to avoid all the toxic stuff. And it's easy to do. Be a low-fat, low-sodium, 100% plant-based, whole food, vegan. That's the smart thing to do. Exercise, get your sleep, and all that stuff. All right. Okay, here's the drawing of it. Hopefully this will help clarify some of these things. So things that increase calcium, high cytoplasmic calcium. So here's Ca2+ inside a brain cell, hippocampal neuron. They're going to activate this calpain protease. Calpain protease is like a pair of scissors that cuts the HNE, hydroxynonenal. Hydroxynonenal has bound to the HSP. The HSP is this little green circle here. That's the protein that stabilizes the lysosome and that also carries these proteins to the lysosome. So it's called a chaperone, in that it guides these old proteins to be recycled to the lysosome. So that's all normal and good. But the disaster is when HNE binds HSP, it targets it for destruction by calpain. Calpain becomes super attracted to it, and it'll destroy the HSP, and then that's going to lead to inability to maintain the lysosome. Lysosome's going to break apart. It's going to release this protease digestive enzyme, cathepsin protease, which is going to auto-digest the cell. By the way, I know this sounds kind of complicated, but I'll tell you, the way to think about biology is just think about it as stories. So good biology creates a coherent story, and that's what I'm trying to do here so you can remember it. In the long run, what's the point? You don't want to eat any cooking oils, not one drop. A lot of people say, oh, I need cooking oils for cooking. No, you don't. You can cook without them. There's lots of ways. Chef AJ will teach you how to do it. You go online, you'll find ways to do it. People didn't even have these cooking oils more than 120 years ago or so. Most stuff you can cook with water. There's other ways to do it. But anyways, all right, getting back to our model for dementia. So other things can increase cytoplasm calcium. If the cell is ischemic, not having enough oxygen, then it can't generate enough ATP to run its potassium sodium ATPase pump, which is coupled to pumping calcium out of the cell. Calcium accumulates in the cell. So what causes ischemia? Well, lack of blood flow. That can be overtreatment of hypertension. It could be congestive heart failure with decreased cardiac output. It could be AFib with decreased cardiac output. Things that increase metabolic demand. Excess glutamate increases the activity of this postsynaptic neuron. Psychological stress, stress equivalents, sleep deprivation, caffeine, for example. All of those things will increase cytoplasm calcium And that means they're going to increase activation of calpain protease. Remember, calpain, it's activated by high levels of cytoplasm calcium. So that's why you don't want that. There's a whole bunch of things that will inhibit this enzyme, the SERCA enzyme. That stands for sarcoplasm, endoplasmic reticulum, calcium ATPase. So the bottom line is the endoplasmic reticulum in a neuron is another reservoir for calcium ions, and calcium is pumped into the endoplasmic reticulum by this SERCA pump, and it helps to lower the calcium level in the cytoplasm. When SERCA is inhibited, and there's a whole bunch of things that inhibit SERCA. I talked about that in my excitotoxin lecture. That will also lead to increased cytoplasm calcium. So you want to avoid all these things that do that, and you just make life a lot easier for your brain neurons. And we talked about the dietary factors, excessive dietary sodium, basically eating processed food. Because the good stuff's in the plants, the potassium and the magnesium, the bad stuff's in the processed food, the really high sodium. So all of these things work together to nickel and dime and destroy your brain cells. And the omega-6 fats, in particular, Yamashima thinks that's the worst of all these things. I'm not sure that's absolutely the worst, but it's up there as being bad. So this is good to be aware of. And I can tell you a lot of people are not at all aware of this. I have a lady doctor, Asian friend. She's, I don't know her exact age. She's young, skinny, smart, pretty, and she thinks, this nutrition is kind of a joke. She says to me, "Oh, Pete, what do you want to do? Live to be 100?" And I just told her, I said, you know what? A lot of people are ALDH deficient. And guess what? They can't handle these cooking oils because she loves eating junk food. Her face really changed when she heard that. So maybe the old man has got something meaningful to say after all. So anyways, I'll tell you another story. I have an Indian doctor friend, real nice guy. He's real skinny. And I didn't see him for a couple months. And so I saw him and I said, "Hey, how you doing? Where you been?" And he said, "Oh, I was dead." I go, "What do you mean you were dead?" And he said, "Well, he had a myocardial infarction. He, in a sense, died, cardiac arrest. He had a code blue. He was resuscitated, came back to life, and then he took some time off to recuperate. He had cardiac stents put in, so that, saved his life. That's great." But I said to him, why would he have a myocardial infarction? He's skinny, seems real healthy, real with it. And he said he didn't know. I said, "Well, do you cook with oil?" And he goes, "Yeah, you have to. You have to cook with oil. How else are you going to cook?" And I said, "No, you don't. That's what caused your myocardial infarction." So whenever you hear a vegan had a myocardial infarction, a health vegan, that's what it is, usually. Okay? Okay, here's some references from the talk. Here's a good paper that summarizes Yamashima's calpain cathepsin theory. So again, that's just activation of calpain either by the binding of HNE toxic aldehydes to the HSP, heat shock protein, can activate calpain to cleave it, and then that leads to lysosomal lysis, or excessive intracellular calcium, cal for calcium, can activate calpain and cause the same process. And people just aren't aware at all of how toxic these omega-6 cooking oils are. They call them vegetable oils. They really come from seeds. They're seed oils like corn oil, safflower oil, sunflower oil, all that stuff, soybean oil, all that stuff. Here's another paper by Yamashima. And then let's see which one I think is interesting. Heat shock protein affects neural fade. Okay. Shower's paper I thought was good for 4-hydroxynonenal, a bioactive lipid peroxidation product. This is a good nutrition and metabolism book by Groper, Advanced Nutrition and Human Metabolism. I like that one. What else? Good here. This is Yamashima's book, which is, his book's kind of so his papers are better than his book. So anyways, hope that's helpful. And the whole point of this lecture was avoid cooking oils. They're toxic to your brain and the rest of your body.